Hi guys, this is Mrs. Foy, and this is a quick screencast for AP Biology talking about control of gene expression in prokaryotes. So we're talking about the Operon system, and this is just a quick little screencast to talk about the CAP system, which is an example of what we call positive feedback control. So that's going to be different than what we see in the Trp and the Lac Operon system, where it's negative feedback control. So I just want to thank Khan Academy because I got these pictures from them. If you go to Khan Academy to their gene expression and regulation section, you will get the tutorial on this, which of course is amazing. Love Khan Academy. So very quickly, and you should already have some background information about the Operon systems, but this is a way that bacteria use systems to control gene expression, to control which genes turn on and turn off. So the Trp Operon system is a repressible operon. That means that normally it is on, but you can turn it off. All right? Normally it is on, the default position is on, and you can turn it off. And so what is happening is that normally this type of operon is going to be present in anabolic pathways. Anabolic pathways is when we take small monomers and we build them together to make a large monomer, right? So we're building. So what is happening here is in the Trp system, we have these enzymes to make tryptophan. And so normally that is going to be on in the bacteria. And so we don't have tryptophan. We have just a little bit of tryptophan. The cell needs to make more. And so the RNA polymerase is going to fit on there just nicely, and it's going to transcribe these enzymes that are needed to make tryptophan. When tryptophan builds up, then it is going to bond to the repressor and make it an active repressor. It sits on the operator, and now the RNA polymerase will not fit on the promoter and no transcription occurs. This is an example of a negative feedback control because watch, when something is on, you can then turn it off. Okay? So negative feedback control does not mean that something is turned off. It means that the result is the opposite of the stimulus. Okay, so because it was on and I turn it off, that's an example of negative feedback. The Lac Operon system, short for lactose, is an inducible operon. This is normally off, but you can turn it on. This is also an example of a negative feedback control of gene expression because the result is the opposite of the original stimulus here, right? So let's look very quickly at this. Lactose is a sugar that the bacteria will use if it has to. It's not its favorite food, but it will use it if it has to, and if it doesn't have enough glucose. And so normally what happens, this is going to be a catabolic pathway because lactose, we know, is a disaccharide. And when lactose is present, I'm going to need these enzymes to break down the lactose into the monomers, okay, like glucose. So when there's no lactose present, we don't need the enzymes to break it down now, do we? And so normally there is going to be a repressor that binds onto the operator portion of the DNA. And that means RNA polymerase can't fit on the promoter where it's supposed to. And so no transcription of these enzymes are going to happen. Well, yeah, because you don't have lactose. You don't need them. But when lactose is present, there is also an isomer. There is an isomer of lactose called allolactose. And that is going to when the allolactose is present, it's going to bind to the repressor. The repressor then falls off because this means lactose is present, and so I'm going to need these enzymes to break it down. When the repressor falls off the operator in the presence of allolactose, which is always there, it's an isomer to lactose. In other words, allolactose is going to be around when lactose is around. Now, the repressor is going to fall off the operator, and RNA polymerase is going to be able to sit on there, and it's going to transcribe these enzymes that are needed to break down lactose. So this is an example of it. This is an inducible operon because it can be turned on, but it is also an example of negative feedback control. But there is a type of positive feedback control that I wanted to talk to you about in regulating gene expression, and this is called the Catabolite activator protein, all right, or CAP. And so what happens is, in this, more CAP, more transcription. And this is kind of an add-on feature to the lactose operon or the Lac operon. So I want to show you about this. But what makes it positive, it's not that it's good. It's just that more CAP means more transcription. So notice in the negative feedback, we always had the opposite. So if we started out with on, it turned off. If we started off with off, it turned on. So these would be an example of positive feedback. I'm sorry, negative feedback. This is an example of negative feedback. In positive feedback, more is going to give you more. Do you see the difference? And so that's what I wanted to discuss. So normally what happens, guys, if you have high glucose present, then those levels inactivate the adenylate cyclase, which is what makes cyclic AMP. And cyclic AMP is going to be one of those molecules that acts in communication. So if you have low glucose levels, then you don't have any inactivation, and so you get lots of cyclic AMP. This is going to be a signaling molecule that's going to start some chemical reactions that happen in the cell. So when you have low glucose, you're going to have high cyclic AMP. So remember, what is the bacteria's favorite food? It's not lactose. Its favorite food is glucose. But it's going to use lactose when it has to. But also, it's going to use the Lac operon when it's low glucose. So let's look what happens here. When glucose levels are low, we have a lot of cyclic AMP that's produced. And cyclic AMP bonds to this CAP protein that sits just upstream from the RNA polymerase site. And when the cyclic AMP bonds to the CAP, it actually helps the RNA polymerase bond really well at the promoter site, and you have high transcription of these enzymes that are going to end up breaking down lactose, which is good because that means that you're probably going to have to use lactose as your sugar, as your fuel. When you have high glucose levels, you have no cyclic AMP. And so the CAP protein cannot bind to the CAP site, and the RNA polymerase is not going to fit as well. You're going to have low transcription, all right? But we're going to see this is going to act in concert with the lactose operon. So when is a lactose operon going to really get turned on? When there is low glucose, all right? So when there is low glucose, we're going to have high cyclic AMP, remember? So that's one thing. And lactose has got to be available. So you have to have lactose available. Otherwise, why would you need enzymes to break it down? So these two events have to happen in concert to get the really high levels of transcription made for that operon. So let's look at this condition. If I have lactose present, and if I have glucose present, but I have no lactose. Well, clearly, why would I need these enzymes? I don't, because I have glucose present. And so if I have glucose present, I'm going to have no cyclic AMP or low cyclic AMP around. So I'm not going to enhance transcription because I don't even need it, right? I've got the repressor that's sticking on here because I don't even have lactose present. So I'm not going to need those enzymes to break down lactose. If I have glucose present and lactose is also present, well, the bacteria are going to choose their favorite food. They really like glucose. It's easier for them to metabolize. So they might have some transcription, okay, of the Lac Operon enzymes, but they don't have a lot because, again, they have glucose present. So if there is lactose present, remember there's also going to be allolactose. It's going to bind to the repressor. The repressor is going to fall off the operator. And the RNA polymerase is going to start transcribing these enzymes that are going to break down lactose. And all that's lovely, but I don't get the enhanced transcription. I don't get the enhanced transcription because glucose is present, and so my cap protein system is not going to work. If I have glucose absent and lactose absent, well, I'm not going to get any transcription. Why should I? Even though I have high cyclic AMP levels, my cap protein is going to bind, but I still have the repressor stuck right here. And so the RNA polymerase cannot transcribe these enzymes because the repressor is blocking it. But when I have glucose absent and lactose present, now I'm going to get strong transcription of the Lac Operon. And this makes perfect sense, right? So when glucose is absent, I'm going to have high levels of cyclic AMP. It's going to bind to my cap protein at the cap site. Also, because I have lactose present, if I have lactose around in the environment where the bacteria are, I'm also going to have allolactose. Remember, it's an isomer. And so it's going to bond right here to the repressor, and the repressor is going to fall off. Now I'm going to be really cooking. So now I have the operator that is open. It's not being blocked. And I have my cap protein activated that's going to really help the RNA polymerase to bind tightly to the RNA promoter. And this thing is going to transcribe like gangbusters, all right? And that's because I need these high levels of the enzymes that are going to break down lactose because I have lactose present and I have low glucose. So this is an example of positive feedback control of gene expression because the cap protein system increases an operon system that is already on. So that's why it is positive feedback. So I hope this has been helpful, and I'll see you.